bioreactor, they perfuse that tube with endothelial cells, and the endothelial cells just sort to the wall, and they create the endothelial layer. That's just a single layer structure, so it's not that difficult to do with the seeding. So what is the underlying science that I so proudly announced? So what I said is that we are really not doing much on our own. We are using some fundamental morphogenetic processes that even in early embryonic development, from early embryonic development, are known. Well, one is that surprisingly, many cell types and many cellular fragments or tissue fragments do behave as liquids. Well, they mimic the behavior of liquids. What does that mean? So you take an irregular tissue fragment. This is a part of an embryonic limb from a chick. You put it on a tissue culture medium, and it just rounds up on its own. Here is the last 7 hours of this rounding process of this same guy, and it does its own on its own. You don't have to do anything to it, and it will round up. This is one particular property of liquids. Liquids try to minimize their area with the surrounding environment. This shows the engulfment of a water drop by an oil drop, and nobody is surprised if you know that at the end of the process the oil drop surrounds the water drop. This one here is 2 embryonic tissue types, and I leave it to your fantasy to compare this structure. It started out like this, and then in this row you can find, to your liking, a configuration that is mimicked by this structure. So again, embryonic tissues, and not only embryonic tissues, many adult tissue types do the same thing. The sorting process that I also mentioned as an important ingredient in this technology is shown here. You mix 2 cell types, and one typically will sort out from the other. Here the colors are the same. So this is liver, here it is liver, this is heart, this is pigmented epithelium, and this is limb, and this is neuroretina, and they sort out in a translational, transitive manner. Or if you change the number of cell adhesion molecules, the same cell type, but one contains, the green one contains 25% more cell adhesion molecules in this case anti-adherence, then they will sort out as well. So what determines physically which one goes out and which one goes in? So you can surmise, as I mentioned here, the cell adhesion molecules, that biologically maybe the cohesivity of the tissue is the one that is important. Now, liquids have a property called surface tension, and this property has been defined by a biologist for those tissues, goes back to Malcolm Steinberg and the end of the 60s. No biologist took it seriously. The idea was taken over by physicists, and now it's hailed as an important contribution to embryonic development. So if this surface tension is measured for those tissues, higher the surface tension, more cohesive the liquid is. And as you can see, this sequence of surface tensions very well reproduces the mutual sorting behavior of those tissues. So you may say, well, all this is fantasy, all this is fortuitous. I don't think that there can be so many factors that are fortuitous. So it is believed today that embryonic development requires a lot of physical mechanisms to act, not only genetic, not only molecular. Genes will not drive physical processes. Physical mechanisms are needed. Genes can give rise to those physical mechanisms, but by themselves, they just don't do that. So here is the final one, the fusion that I mentioned. Here are 2 embryonic cushion tissue fragments that fuse. I talk too much. These 2 movies show the first difference between the true liquid and the tissues, at least glance into the aggregate. There is adhesion-driven fusion and there is diffusion-driven fusion. This one lets the units to spread in a uniform manner. This one does not. There is just an interface, but both of them round up. They both minimize their area with the environment. And the rounding process or the fusion process can be described again with the theory of liquids, which is amazing. So this is a kinetic process and still can be described. So this is what essentially we use. Before I go on, I mentioned embryonic development. Here is one example. This is a 10.5 embryonic day mouse. And what you see here is the pair of dorsal aortae on this mouse. And those are DIC images. And those two guys, the two channels, those are really two channels, two cylinders, they approach each other in the course of development and eventually fuse into one. So this is a very similar process to the one that we're using in our engineering technology. So let's go over to the outcome.